Okay, so Jean-Francois has given a nice introduction to the financing and the fundraising behind DNDi. I'm going to tell you a little bit about our model and tell you about some of the innovations that we're applying, particularly in the open space, to try and help, particularly in early research. So just a reminder, DNDi is a not-for-profit virtual drug discovery organisation focused very much on patients' needs for the most neglected of diseases. You can see the diseases for yourself here, and what keeps us true to our purpose is having very clear, well-defined target product profiles that we publish on our website. This is good for keeping us on track, but I think it also helps us enormously with our partners in recruiting others to work in this field. There's a very clear goal that we're all agreed on, and it's really rooted in the needs of those patients. DNDi is 10 years old. It was 10 years old last year. It's been, I think, pretty successful, and just since 2007 it's managed to launch 6 new treatments, and as Jean-Francois said, it's this changes in the impact on research for neglected tropical diseases and using the investments that have been made well to give some more impacts like this is the key for us in the next 10 years. So DNDi has a reasonably busy portfolio, not least because we work across a panel of diseases. As you all know, drug discovery is a risky business, and although there's lots of compound names and numbers in the boxes here, many of them sadly are destined to fail. So it's really important for us to work hard to keep refueling the pipeline and to make sure that there are future agents to go into the clinic. DNDi is obviously, like the other operators in this field, keen to share and use open data as much as we can. What does this look like practically for us? Well, we publish like everyone else, so there's a ream of papers for fexinidazole, for example, talking about its repurposing discovery, its development, and now into the clinic. That's great, that's quite traditional, it's a very important part of what we do. We're also trying to deposit data earlier and a more comprehensive deposition of data into databases such as Kemble. So our screening data and our optimization data, as much as possible, we'll put that into the open domain so that other people can use it. We benefit from lots of other initiatives. We contribute to others. One I'd pick out here is the pathogen box. We think this will be a really exciting step forward from MMV's malaria box. I think that the initiative to spread this out to other pathogens is really important. Should foster a lot of basic research into targets, pathways, repurposing, and all sorts of other things that are going to help across a spectrum of diseases. So DNDi is a keen supporter of this initiative as well. But I think it's fair to say that open innovation means a different thing to everybody on the planet, me included. I'm not an expert in the open source, open innovation definitions and criteria. For me, it comes back to something a little bit more, a bit more basic. I'm a bit more of a reductionist. Science for me works through sharing and collaborating. There's a continuum of more or less open approaches, but I think it boils down to 3 questions. What's shared, when's it shared, and with whom's it shared? You can wait 5 years for the Nature paper, but that's going to stifle the rapid follow-on and use of that information. You can put all your results in the open domain from the start, but that obviously brings some challenges with it around IP and funding and publishing and everything else. So I'll let others worry about exactly what the title is and think more about what can we do to change the status quo and to tackle some of the issues that we face in early research. And I'm not going to try and share everything straight away with everybody because it's probably not going to be useful anyway, and it will kill me trying to do it. I'm going to focus on the things that I think will be most impactful. And I'm going to tell you about some of the bottlenecks as we see them in the R&D pipeline, and particularly focusing in the research end where I operate. So what we characterise as research being high throughput screening, identification of hits, going into the hit to lead process and the lead optimization process, such that we can present new preclinical candidates to go on further into development. So I've just picked 3 areas that I want to pick up on. We have quite a bit of data ourselves. There's quite a bit of published data. And I think a good question is, do we always use that data as intelligently as we could? Do we see the whole picture when we're thinking about chemical series and their optimization. So have we done everything we can to know our molecules? And given all that information, what more can computers do for us in terms of building predictive activity models, helping us to select further starting points and optimize the ones that we've got? So using that data will be one theme. The main project I'm going to tell you about is the NTD Drug Discovery Booster. So following up on the question I asked before lunch, DNDi spends a lot of time working in very exciting and effective bilateral relationships with a number of pharmaceutical companies. And this is an important pillar of what we're trying to do. But it does take us time, and quite a lot of those activities necessarily are partitioned. So we'd very much like to make that more of a collaborative environment in which we start to work with several companies together and share a little bit more information and resources. So I'll tell you some more about that. And then finally, once we get projects running in lead optimization, we spend quite a lot of time and money and resources to do that medicinal chemistry optimization with all the partner disciplines that are required. And so we're looking at new ways to harness existing resources and to bring new resources into the neglected arena. So our lead optimization Latin America project, LOLA, and some more open chemistry partnerships that we're planning to bring forward will be the last part of the talk. So moving on, drug discovery, particularly for kinetoplastids, so the parasites that cause HAT, Chagas and leishmaniasis, are tricky things to find hits for. Particularly for the intracellular parasites that cause leishmaniasis and Chagas disease, the hit rates are exquisitely low, less than 0.01% in primary high throughput screening. So although the throughput's increased in recent years with industrialisation and the efforts of organisations such as GSK, we still don't have enough hits to really be confident that we're going to deliver the multiple candidates that we need if we're going to go through and deliver more than one NCE to give us combinations of oral drugs for the patients. So it's really important for us to do more to find more hits, but when we get a hit, we've got to do everything we can to make the most of it, make sure we pick the right hits and we move them forward quickly. And this comes to my first point. So we've got some data, there's some published data, and we want to make sure that we've captured this and we're using it as well as we can, so that when we start to make important investment decisions about chemistry and starting projects, we're doing it in the right place, we're addressing the right issues, and we're moving as quickly as we can. And given these precious hits, can we use them to find others? So can we build computational models? Can we go and look in commercial collections or in pharmaceutical companies' collections in a more guided way rather than high throughput random screening, which takes a lot of time and costs a lot of resources? So the first of these is recognising that DNDi screened 15 or 20 organisations multiple times, many compound collections, and we've reviewed these on an individual basis. But to take a step back and look at all of that data, and look at all the data that's in the public domain in databases such as Kemble, so that we can really annotate those compounds as individuals and as series and cluster them together, has allowed us to develop a workflow where we've gone through and reanalysed all of these hits, reviewed them and identified some series that were previously unknown to us, perhaps because they were made up of singletons from different screening campaigns. We've then been able to go and apply searches to public databases, commercial compounds, purchase these and test them to see if we can expand the SAR, and we'll be able to go further by sharing these models with our pharmaceutical partners as well. But all of this work is only as good as the data that we have. So the more data people publish and deposit or give us access to, whether it's in an open sense or whether it's in a shared confidential basis, the better we'll be able to do this job. And just to think about predictive activity models, we're looking for needles in haystacks, and if we're going to do something better than sticking our head in a haystack and ferreting around for ages, it would be much better if we could be a bit more guided. Could we use a magnet to pull the needle out of the haystack a bit quicker? So again, this is building those models, 2- and 3-dimensional models in computers, so based on fingerprints, based on 3-dimensional structures. We've then made predictions of activity of compounds in commercial libraries. We've purchased those and we're in the process of screening them. We're optimistic that this will give us some new starting points. and that given the validation of these models, to then use them with our pharmaceutical partners in their really large and interesting diverse collections of compounds will help us find some more of those precious needles in the haystacks. The data sources that we've used for this activity have come from some companies, so AbbVie, GSK, some organisations such as the Dundee Drug Discovery Unit, from Institut Pasteur in Korea, and from published collections. But it's still only a fraction of what we know has been generated, and so it's still a major goal for us to try and access more of this, to encourage people to publish it and share it, because the more data we have, the better the results will be. However, I think the conclusion from this is that even a bit of the data that and publishing more of the data can be very enabling and allow us to do some really valuable activities in discovery. So same challenge, different approach. So what else can we do to make really good use of the hits that we have from high throughput screening? And this is where the neglected tropical disease drug discovery booster concept comes from. So that the aim is to expand the hits that we get from screening quickly to enable us to find related series that are perhaps a slightly different scaffold, and also to annotate those series, so get information from different partners that have worked on those series and help us to make better decisions and to guide the forward program better. And overall, the intention is that this will make it faster and reduce the cost of discovering new clinical candidates. So what does this look like for the chemists in the room? We might find a chemical structure from a high throughput screen. Probably for some of the difficult parasites, we might have to screen tens or hundreds of thousands of compounds to find one series that we think is really valuable. So we want to make the most of this seed. Traditionally, chemists would go into the lab and make lots of analogues, and that takes a lot of time and a lot of money. Each of those analogues could have a subtle modification in the structure, and that helps us to understand the relationship between structure and activities. However, there's a good chance that a lot of those compounds have been made somewhere else on the planet before, and we know that the pharma industry has millions of compounds, and within those millions of compounds, probably some of these analogues. And so we'd really like to work with the partners that have the different pieces of the picture, of the puzzle, to put it together with us. So rather than having to make all the compounds afresh, or just working with one organisation that's perhaps got a few analogues, we'd like to work across the industry, or at least with a significant portion of it, to build the picture more quickly and more efficiently. The analogy again, we want to look in multiple haystacks. We don't want to do it one at a time. We want to do it all together with a really big magnet, and to try and pull out the best hits and the best annotations that we can right up front. So just to try and summarise that, the goal is faster, cheaper drug discovery, that rapid expansion of a new hit. Crucially, we'd gain a lot of information and develop SAR before we have to commit to new chemistry. Chemistry takes time, takes resources, it costs us a lot of money. So if we can use existing information, existing compounds, it's much better. And ultimately, many of these series will have been worked on as part of programs with inside companies, and the information they can give us about those series may be overlapping, it may be different, but it will all be valuable, and it will really help us to move those projects forward, to select the best projects and then to move them forward with the most information and the best guidance possible. So those annotations tell us about the good things, but they also highlight the risks for us as well. So if there's a series that's just too risky, we won't start, and if there's a critical risk, we can address it early. So the annotations are a really valuable piece, and the advice and the guidance from the pharmaceutical partners that have worked on these series will be invaluable. So without diving into too much detail, what does the NTD booster look like? So here's quite a complicated diagram, but we would aim to start on the left-hand side with a single compound, as I showed you a couple of slides ago. This would be the seed for growing the series. We'll show this structure to a number of companies. I've shown 4 here. I hope it'll be more. And that each of these companies will then go and use computers to search their corporate collections and pick out 100 most similar compounds to our original hit from their 1, 2, 3 million compounds that they may have in their collections. DNDi will then screen them. This will all be blinded to the other members of the consortium, but we'll see, we hope, better compounds coming out of this screening process. And the very best compound that comes out of that iteration across the whole consortium will provide the hit for the next iteration. And we'll run this process in cycles. We'll iterate it perhaps 2 or 3 times to really mine out the best of the compounds and as much of the SAR as we can. We hope this will give us a better series with more examples, And crucially, at this stage, we'll really be asking for as much annotation as we can from the partners. So we'll have a series where we've got lots of information, extended SAR, and a good idea as to whether this is a series we can take forward, and if it is, what should we be addressing, and maybe how to address those issues. So overall, we hope this would reduce the time for hit to lead, which is a significant portion of our current research process, from perhaps 12 to 18 months down to 3 to 4 or 6 months. And that because we'd be starting in a stronger position in lead optimization, we might halve the time it takes us to do lead optimization. This is a difficult, resource-intensive and expensive stage of the process, so this will be a major cost saving. It might take us 4 to 5 years and 5 to 6 million euros for the sake of a starting point to complete the research phase, to go from screening hits through hit to lead and lead optimization. We hope that the booster will significantly reduce the hit to lead component of this process in terms of time, and also speed up the lead optimization process. And so this critical work with across the private sector, we hope will really reduce the time and also the cost to do this early research phase. And let's face it, doing early research is the most difficult piece to raise funding for, because we're furthest away from the patient. So this is really important for us to manage our own shop and try and speed up and reduce costs. Furthermore, we're going to work more to speed up the lead optimization process. This is where we're starting to do a lot of chemistry. There's a huge capacity for doing synthetic and medicinal chemistry within Europe and across the globe. Only a small portion of it is applied to NTD research at the moment, and we're very keen to work in a more open way to engage particularly some more of the public sector and some more universities to help us move those chemical series forward once we've identified some good starting points. And we're hoping that this will further drive the costs down, and I think critically increase the sustainability of the process. So to keep having to go back to funders to raise money for early research becomes increasingly challenging. If we can build a more sustainable model where we're really engaged with the academic world as well as the public, as well as the private sector, I think we'll have a more sustainable model for future drug discovery. So this is kind of a picture of where we want to go. There's a couple of other things I've mentioned in my talk. The pathogen box, which we hope is going to give us more choices for screening and better choices for the hits that we start on, and tell us about the targets and pathways that we screen in. So DNDi is a keen supporter of this, which is clearly at the very open end of innovation. And I mentioned briefly our lead Latin America consortium. So again, this is a way to try and move forward series in chemistry, moving into an area in Brazil where there's an enormous scientific capability and capacity, huge will to work on diseases that are endemic in Brazil and Latin America, and with some extra input from DNDi and our partners, we hope to really harness some of those resources again in an open way to help us move these projects forward to discovery of new clinical candidates. So I hope that gives you a snapshot of where we see some of the pinch points in early research and what we're trying to do to try and address some of them, particularly with more or less open projects, but certainly with science and with collaborative approaches. So I haven't got a slide with all the partners that we're working on because there are so many, but thanks to all of them, to the donors, the scientific partners, public and private, all the institutions that we work with, and to you for your attention.